Yeah, good morning to all the third year PharmD students. So this is Dr. Sunil Kumar. So in the last class, we have discussed about cotrimoxazole. So we have seen how effective the combination of sulfamethoxazole and trimethoprim, which is called as cotrimoxazole, how effective the combination is. So individually, these two drugs, sulfamethoxazole and trimethoprim, they are bacteriostatic. But when you combine, collectively, they act as bactericidal. Okay, and these two drugs will act at different sites of synthesis of folic acid from PABA. Okay, so sulfamethoxazole will block the conversion of PABA to dihydrofolic acid by inhibiting the enzyme folate synthetase. Okay, and trimethoprim, which is a diaminopyrimidine and whose structure is similar to an anti-malarial drug, that is what we call it as pyrimethamine. So this trimethoprim, it is going to inhibit the enzyme dihydrofolate reductase, and it is going to block the further conversion of dihydrofolic acid to tetrahydrofolic acid. Hence, if any bacteria tries to synthesize, or if any bacteria develops resistance to sulfonamides, and if it converts PABA to dihydrofolic acid, okay, then you have another drug here which is going to block the conversion of dihydrofolic acid to tetrahydrofolic acid. Hence, so individually these drugs are bacteriostatic, but collectively they are bactericidal. And as I told you, they work by dual mechanism. So there is this dual mechanism will provide some minimum risk of bacterial resistance. Okay, that is what we discussed. And now I would like to discuss about the well-known antibiotic, which is the first antibiotic discovered, that is none other than penicillin. So let's begin with penicillin, and we will continue with other beta-lactam antibiotics. Okay. So followed by, we will discuss the other antibiotics like tetracyclines, aminoglycosides, fluoroquinolones, etc. Okay. So let me share a screen. Just a minute. Yeah. So this is about penicillins. Just a minute. Let me zoom it a bit. Yeah. So when talking about antibiotics, let me remind you something. See, what we discussed, the sulfonamides and cotrimoxazole, they are not antibiotics. They are antimicrobial agents, or you can call them as chemotherapeutic agents. Why? Because in the beginning of the discussion, while beginning chemotherapy, I clearly defined what is antibiotic and what is chemotherapeutic agent. Antibiotics are drugs which are obtained from microorganisms, okay, which has to be produced by microorganisms and which are used to kill other microorganisms at very low concentration. Okay, when we use the word antibiotic, means it should be derived from microorganisms. Whereas sulfonamides and cotrimoxazole, what we discussed, they are not derived from microorganisms. They are derived by synthetic procedures in the laboratory. That's why you can call them as antimicrobial agents, or you can call them as chemotherapeutic agents. Okay, so you can't call them as antibiotic. Okay, now let's come to the antibiotics. So in antibiotics, the drugs which are going to inhibit or kill microorganisms, so which are derived from microorganisms. Okay, so that is in that the first category I would like to discuss, that is beta-lactam antibiotics. Okay, beta-lactam. So these drugs will have a beta-lactam ring. These drugs will have a beta-lactam ring. I think if you observe this slide carefully here, so I would like to write here. So this is the beta-lactam ring, CH dash CH C double bond O. Okay, and here this is N. This is N. Okay, so this is this kind of beta lactam ring drugs having this kind of beta-lactam ring are called as beta-lactam antibiotics. Okay, so as the definition says, these are the antibiotics having a beta-lactam ring. So these are the antibiotics having a beta-lactam ring. And here you have two major groups. One is the well-known penicillins, and next the drugs which which will have a similar mechanisms like penicillins, but they are somewhat better than penicillins. Okay, they have minimum side effects and they have broader spectrum than penicillins. They are nothing but cephalosporins. Cephalosporins. Okay, so beta-lactam antibiotics are the antibiotics having a beta-lactam ring, which I have shown here. These antibiotics are divided into major two groups. One is called as penicillins and another one is cephalosporins. And you have two new additions to these beta-lactam antibiotics. One is called as manobactams and another one is carbapinum. So manobactams and carbapinums are the latest additions or the newer additions. Okay, so I will come to that. So now let's begin with penicillin. Yeah. So this is penicillins. So penicillins, as you know that penicillin was the first antibiotic discovered by a scientist, well-known scientist, that is Alexander Fleming in the year 1928. In the year 1928, Alexander Fleming discovered the penicillin. And originally penicillin was obtained from fungus. You please concentrate here. As I told you, antibiotics has to be produced or derived from other microorganisms. So penicillin is an antibiotic which is derived from fungus that is called Penicillium notatum. Penicillium notatum is the fungus from which penicillin is derived. Earlier, originally it is obtained from Penicillium notatum, but the current source, whatever the penicillin right now we are getting, that microorganism or that fungus is not Penicillium notatum, it is Penicillium chrysogenum. Chrysogenum, chrysogenum. Chrysogenum is a mutant strain. Okay, it is a mutant strain of Penicillium notatum. Okay, so certain mutations has been created to Penicillium notatum, so that that mutated fungus is going to produce a large amount of penicillin. Okay, it is going to give a large high yield of penicillin. So that's why that mutated species of Penicillium notatum, which we call it as Penicillium chrysogenum, it is used right now in order to produce penicillin. Got it? So earlier the original source is Penicillium notatum, but right now the current source is, or the present source is, a mutant fungus of Penicillium notatum, a mutant species of Penicillium notatum, that is Penicillium chrysogenum, which is going to give high yield of penicillin. Okay, so current source is Penicillium chrysogenum, original source is Penicillium notatum. Okay, got it? So that's it. So that is about the introduction. Now coming to the chemistry, if you just go through the chemistry, I think this structure is quite visible to everyone. So you can see that penicillin is a fused ring. Okay, it contains two fused rings. I think this portion, which I am highlighting in this yellow color, so this portion is called as thiazolidine ring. Okay, so this is thiazolidine ring. Please try to understand. This is thiazolidine ring, and which right now I would like to highlight in some other color. Let me just a minute. Let me take this red color. So this particular ring, okay, which I have highlighted in red color, please observe. So this ring is called as beta lactam ring, which I have shown here. So this is beta lactam, beta lactam, Lactam. Okay, so here, so this yellowish portion which I have shown you, that is thiazolidine ring, and this red colored portion which I have encircled, so that portion is called as beta lactam ring. Okay, so penicillin is made up of two fused rings. One is thiazolidine ring and beta lactam ring. Okay, and you can see here there are side chains attached. This one particularly, this side chain, this is nothing but amide linkage. Okay, so let me read it out here. You can see penicillin nucleus, it is consist of fused thiazolidine and beta lactam ring. It is made up of fused thiazolidine ring and beta lactam ring, and to which side chains are attached through an amide linkage. You can see here. So this is an amide linkage. Okay. So this is an amide linkage. I will show it here. So this particular portion. Yeah. So this is an amide linkage and this R is the side chain. Okay. You can add side chain to the penicillin through this amide linkage. Okay. So side chains will be added to this fused beta-lactam and thiazolidine ring, which we call it as penicillin. This side chain will be added through this amide linkage. Through this amide linkage. Okay, and here you can see how penicillin will be further broken down. You can see here. Let me before that, let me just discuss the types of penicillin here. You can see here, 4 types of penicillins are naturally available. One is called as penicillin F, penicillin G, penicillin X, and penicillin K were produced originally, but the penicillin G, which is obtained, as I told you, obtained from Penicillium chrysogenum, okay, mutant strain of Penicillium notatum. So that penicillin G, okay, it is having a benzyl side chain. It is having a benzyl side chain, and this penicillin G, whatever right now we are using penicillin in most of the hospitals, so it is nothing but natural penicillin, that is penicillin G, and which is having a most desirable properties, which is having most desirable properties, and it is used, as I mentioned, in most of the hospitals and clinics. It is used clinically. Okay, so that R which I have shown you, if this R is replaced by a benzyl side chain, okay, so you replace this R with a benzyl side chain. Okay, in place of R, if this C6H5CH2, which we call it as benzyl side chain, you replace R with benzyl side chain, then it becomes penicillin G, which is having more desirable properties and which can be used clinically. Okay, so that's it. And here you can see penicillin G, which can be written as PNG. You can write it in the short form as penicillin G. PNG. It is thermolabile acid labile. Means it is sensitive to temperature and it is sensitive to acid. It is sensitive to acid. So that's why penicillin G is not directly given orally, because if you take a tablet of penicillin G directly through oral route, so most of the penicillin G will be destroyed in your gastric acid of the stomach, in your acidic environment of your stomach. That's why penicillin G will be given in the form of intramuscular injections. Okay, I think most of us in childhood we have penicillin G. Okay, unless and until you are allergic to penicillin G, so most of the times we use penicillin G. Okay, in most of the infections. But now we have better drugs like cefixime, cefodoxime, cefotoxime. So we have amoxicillin with clavulanic acid. So we have wonderful drugs. But earlier in 90s, penicillin G was widely used. Okay, right now also we are using, because penicillin G is very effective in many infections. Okay, so even with these advanced drugs what we have right now, penicillin G is most effective in few infections. Suppose in case of meningitis infections, in case of severe streptococcal infections, penicillin G will be used immediately. Okay, so but the problem is it is thermolabile, that's why injections are preferable. Sorry, it is acid labile, that's why injections are preferable. Oral route, it is not preferred. Okay, and it is thermolabile, it should be refrigerated. It should be refrigerated because it is very sensitive. It immediately deteriorates at higher temperature. Okay, so that's it. And here you can see the side chain of penicillin G. It is split by an enzyme that is called amidase. It is split into 6-amino penicillinic acid. Okay, I will show you here. So this side chain, okay, NHCO, this side chain what I have shown you in this black color, NHCO, and here you can see the arrow mark Y. Y. So Y is an enzyme that is going to break this NHCO bond. This NHCO bond will be broken by an enzyme that is called as amidase. Amidase will break that NHCO, and that will result in the formation of 6-amino penicillinic acid. 6-amino penicillinic acid, 6-amino penicillin acid. Okay, and there is also another enzyme. You can see here, yeah, here this beta lactam ring. Yeah, here, this beta lactam ring here X. It has been shown as X. Okay, so this beta lactam ring will be broken down by an enzyme which is shown here as X. So this enzyme X is nothing but penicillinase. Penicillinase is an enzyme that is going to break down beta lactam ring and that is going to inactivate penicillin. That's why in many cases of resistant bacteria, okay, back one of the mechanism by which bacteria gains resistant to penicillin is they produce penicillinase, and penicillinase will inactivate this beta lactam ring and it is going to inactivate the penicillin. Okay, that's why if you just go to any medical shop or hospital, you have drugs like penicillin plus cloxacillin, penicillin plus dicloxacillin, penicillin plus neficillin. So what are these cloxacillin, dicloxacillin and neficillin? Generally they are given with in combination with penicillins. They are called as penicillinase resistant penicillin, or I use the word penicillinase inhibitors. They will inhibit the penicillinase and they will prevent the breakdown of beta lactam ring. They will inhibit the breakdown of beta lactam ring by penicillinase, and thereby they are going to protect the beta lactam ring of the penicillin, or in other words, they will protect the inactivation of penicillin. Okay, so that penicillin can further inhibit gram positive, many gram positive organisms. Okay, so that's why what I am saying, uh, 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 many in many cases where penicillinase is produced by resistant bacteria, we have to block that penicillinase. We have to inactivate that penicillin. Sorry, we have to inactivate that penicillinase enzyme so that it will not damage our antibiotic. Okay, that's why. So we have drugs like oxacillin, cloxacillin, dicloxacillin. Sorry for the disturbance. There was some network issue. Let me share the screen again. Yeah. So what I was saying is penicillinase is an enzyme that is going to inactivate penicillin antibiotic by breaking the beta lactam ring. Okay. So it cleaves the beta lactam ring. That's why if you want to protect your penicillin, you have to inhibit that enzyme penicillinase. That's why we are using drugs like oxacillin, dicloxacillin, neficillin, okay, which will inactivate the enzyme penicillinase and which will protect the penicillin, okay, which will protect our antibiotic penicillin. So I will discuss that when I discuss about semi-synthetic penicillins. Okay, so here penicillin is vulnerable, or it is going to be inactivated at it is going to be broken down at two sites. One I told you at this side chain amide linkage, this will be broken down by an enzyme called amidase, and penicillinase beta lactam ring will be broken down by an enzyme which is called as penicillinase. Okay, and a few other properties of penicillin, let me also quote here. Okay, so here it is written in very small letters. Let me just read it out. Let me just highlight. So other side chains can then be attached to it, resulting in different semi-synthetic penicillins with unique antibacterial activities and different pharmacokinetic profile. Okay, so here this R position to this amide linkage, you can add many side chains which will give different penicillins, with unique antibacterial properties and different pharmacokinetic profiles. Okay, for example, you will have semi-synthetic penicillin like amoxicillin, ampicillin, dicloxacillin, which will have a longer duration of action, which will also have unique pharmacokinetic profiles, whose half-life will be longer, whose duration of action will be longer, and whose spectrum is also longer. So such kind of new generation of penicillins will be produced if you add side chains at R position of this amide linkage. Okay, you will get many different penicillins which will have unique antibacterial properties and different pharmacokinetic profiles. Okay, so that I will come to that. So that is one thing. And here, if carboxylic group attached to the thiazolidine ring, okay, so if that Yeah, again, there was a network issue. Sorry for the disturbance. So let me continue from where I was discussing. So please try to understand. See what I'm saying here, just a minute. So I was saying that if this is CO, if this is CO, just a minute. Yeah. So if this carboxylic group attached to this present in the beta lactam ring, this CO, to this CO, if you attach some, I meant to say sorry, if this carbonyl group, sorry, I am extremely sorry sorry, here, this COO, am I right? So this is COO, carboxylic group, this COOH, am I right? So if this COOH, to this COOH, if certain salts are attached to this COOH, like sodium is attached, or if you attach salt like potassium, potassium is attached, K plus, okay, so it will become COONA or COOK, okay. So to this carboxylic group COOH, if you add sodium or if you add potassium, that results in the formation of sodium penicillin or potassium penicillin. And what is the advantage of this sodium penicillin and potassium penicillin? Let me tell you, sodium penicillin G is highly water soluble. It gives more water solubility and it gives more stability. It gives more water solubility and it gives more stability. Let me repeat this. If sodium and potassium occur are attached to this carboxylic group, then there is a formation of a salt. Let me come back to the discussion. Yeah. What I was saying, if to the carboxylic group of the thiazolidine ring, if you add sodium or potassium, then there is a formation of salt formation occurs. That is sodium penicillin G or potassium penicillin G can be synthesized. And what is the use of this? What is the advantage of this conversion of penicillin G into sodium penicillin G and potassium penicillin G? Let me tell you, as I told you, there are two major benefits. One, their stability will be enhanced, and one, if you convert it into a salt, if you convert penicillin G into sodium penicillin G or potassium penicillin G, definitely stability will be increased and their water solubility will be enhanced. Okay, so salt form of penicillin G, which is sodium penicillin G or potassium penicillin G, will be highly water soluble when compared to penicillin G. Okay, when compared to a normal penicillin G. Okay, so if you convert it into salt, there is an improved stability and there is an increased water solubility. Okay, and let me also tell you, when these salt solutions, what I just now said, penicillin G, sodium penicillin G or potassium penicillin G, they should be freshly prepared. Okay, so just before the administration, just minutes before the administration, you have to prepare. Otherwise, if you prepare penicillin G and if you refrigerate it for a long duration, then definitely it deteriorates. It becomes unstable. Okay, that's why these penicillin preparations has to be prepared freshly just before the injection. Okay, so that's it. So that is what the details of penicillin G. Okay, so the freshly prepared solution is more stable than the previously prepared penicillin solution. That's it. Okay, that's why penicillin preparations has to be prepared freshly, otherwise the solution will deteriorate. Okay, yeah. Whenever it is needed, you prepare a fresh solution and inject it. Okay, that's it. So now coming to the most important aspect, that is mechanism of action. So how this penicillin is going to inhibit bacteria, or how this penicillin is going to kill the bacteria. Okay. So here, that is, first I would like to say that penicillin is a bactericidal antibiotic. Okay, means it is not going to just inhibit the multiplication of microorganism. It is going to kill the microorganism. Okay, it is going to completely kill the microorganism. That's why it is bactericidal. Okay, it is not only going to reduce the count of bacteria, it is also going to kill the bacteria. Okay, that is the first point. Next point is, it is going to inhibit cell wall synthesis in bacteria. So that you should understand. It is going to it is going to inhibit cell wall synthesis. Cell wall synthesis. cell wall synthesis. Okay. So to understand this, you should understand, first you should know what is the cell wall of the bacteria made up of. Okay. How the bacterial cell wall appears and what is the chemical composition of the cell wall. That is very essential to know. So let me show you a diagram. So I think this is quite visible to everyone. This is the gram positive cell wall. Okay. So this is the structure of gram positive cell wall. And here you can see this gram positive cell wall is made up of major two components. If you observe carefully here, you can see NAG. Okay, NAG. And here you can see NAMA, NAMA and NAG. Okay, you can see everywhere you have NAMA, NAG. So these are the two components with which bacterial cell wall, especially gram positive cell wall, is made up of. That is called as N-acetyl glutamic, N-acetyl glutamate, N-acetyl glutamate and N-acetyl muramic acid. Okay, you can call it as N-acetyl glutamic acid or N-acetyl muramic acid. NAG is nothing but N-acetyl glutamic acid and N-acetyl muramic acid. Okay, the same thing I have shown here, you can just observe. Okay, so this is NAM, NAG, NAM, NAG. Okay, so these are the two major components of the cell wall. That is what we call it as N-acetyl glutamic acid and N-acetyl muramic acid. Okay, N-acetyl glutamic acid and N-acetyl muramic acid, which is NAG and NAMA, it is the composition of the gram positive cell wall. Then what's next? So the thing is, these N-acetyl muramic acid and N-acetyl glutamate, so they are present in each and every layer. Okay, of the cell wall. You can see here, this is one layer, this is another layer of N-acetyl muramic acid and N-acetyl glutamate, or here N-acetyl glutamate and N-acetyl muramic acid, third layer. And these layers of N-acetyl muramic acid and N-acetyl glutamate, they are tightly held with each other by a process called as cross-linking. You can see here. This is called as cross-linking. Cross-linking. Okay. So this cross-linking is very, very essential to hold these chemical components, which is nothing but N-acetyl glutamate and N-acetyl muramic acid. So these two components, you can see here, NAMA of this is tightly held with NAMA of the above layer. And you can see here, okay, so here also, NAMA is tightly held to NAMA here. So these layers will be tightly held with each other by a process called as cross-linking, and this cross-linking is very, very essential. If you don't have the cross-linking between this particular layer, this particular layer is called as peptidoglycan. Please try to understand. Okay, so peptidoglycan, this complete layer, what I have shown here, though this is called as peptidoglycan layer. Okay, this N-acetyl glutamate and N-acetyl muramic acid and alternate components which are arranged in a sequence. So that entire layer is called as peptidoglycan. Please try to understand. Peptidoglycan layer is the major component of the cell wall. If anybody asks you, so what is the gram positive cell wall made up of? The first sentence that comes that should come from your mouth is peptidoglycan. Peptidoglycan is the layer with which is the major component of gram positive cell wall. Glycan, GAN. So peptidoglycan. Okay. So these peptidoglycan layers above and below, they are tightly held with each other by cross-linking. And you can see here, what are the components of cross-linking? Many amino acids will be there. L-alanine, this is L-alanine. Okay, and this is D-glutamic acid. Okay, and you have this one. This is the DAP, diaminopimelic acid. Diaminopimelic acid. And here, this is peptide bond. Here you can see, this is D-alanine. This is D-alanine and this is peptide bond. Here, this is the peptide bond. Okay. So, 4 amino acids, L-alanine, D-glutamic acid, diaminopimelic acid and D-alanine, they are cross-linked with each other by a peptide bond. And let me tell you, this cross-linking is very, very essential for the rigidity of the cell wall. So both the peptidoglycan layers above and below, they are very, very rigid. They are very, very strong. Only if the cross-linking is perfect. Okay. And this cross-linking is done by amino acids like L-alanine, D-glutamic acid, diaminopimelic acid, D-alanine, and peptide bond. You can see here also. So these four are tightly cross-linked by these four by this peptide bond. Okay. And this will maintain the structural integrity of the gram-positive cell wall. So without this cross-linking, the cell wall will not be rigid. The cell wall will not be strong. And now this cross-linking is mainly characterized, or it is mainly done by an enzyme that is called transpeptidase. Okay. So this cross-linking is mainly done by an enzyme called as transpeptidase. Transpeptidase. Sorry, this is D-A-S, transpeptidase. Okay, so transpeptidase is an enzyme that is responsible for the cross-linking of the peptidoglycan layers in gram-positive bacteria. Okay, I will just revise what I have discussed so that I can come to the conclusion. So what I was discussing is gram-positive cell wall. First of all, it is made up of many peptidoglycan layers. So as I have shown you here, so I have circled, this is one peptidoglycan layer, this is another peptidoglycan layer, and this is another peptidoglycan layer. Now, what are the components of the peptidoglycan layer? So that is also clear here. One is N-acetyl glutamate or glutamic acid, and another one is N-acetyl uramic acid. N-acetyl uramic acid and N-acetyl glutamic acid are the major components of the peptidoglycan layer. Okay, fine. And these peptidoglycan layer, they are tightly held with each other by cross-linking with several amino acids like L-alanine, D-glutamic acid, diaminopimelic acid, D-alanine, and peptide bond. Okay, so this layer, it is going to be held tightly. So these two peptidoglycan layers are held tightly by each other by different amino acids with a peptide bond in between. And this cross-linking, which enzyme is mainly responsible for this cross-linking? So that is transpeptidase. Transpeptidase, and in some books they will use the word PBPs, penicillin binding proteins. They are also called as penicillin binding proteins. Okay, transpeptidase are also called as penicillin binding proteins. Please try to understand. Now, what our penicillin will do? Let me just tell you how the penicillin is going to act. I will write here penicillin G, PNG. So what this penicillin G will do? Penicillin G will directly, I will show you a minus sign. So penicillin G will directly inhibit the enzyme transpeptidase. Penicillin G will inhibit the enzyme transpeptidase. You can imagine what will happen. If transpeptidase is inhibited, can we expect this cross-linking? No. Cross-linking between peptidoglycan layers with the help of amino acids like L-alanine, D-glutamic acid, diaminopimelic acid, D-alanine, and peptide bond. So the cross-linking will not occur. When there is no cross-linking, can we expect the peptidoglycan layers to remain rigid and strong? No. So peptidoglycan layers will become weak in the absence of cross-linking. Okay, and cross-linking is due to transpeptidase enzyme or penicillin binding proteins. So what the penicillin G will do, it will bind with the transpeptidase or penicillin binding proteins enzyme, and it is going to inhibit the function of transpeptidase or penicillin binding proteins, and they will prevent the formation of cross-linking. The enzyme function is to carry out the cross-linking. So the cross-linking does not occur between peptidoglycan layers, and when there is no cross-linking, the peptidoglycan layers become weak. The cell wall which is synthesized, it is made up of weak peptidoglycan layers, which is going to collapse, which is going to break, and finally the cell wall will break down, and that will result in leakage of cellular contents, or that will result in I mean to say that hyperosmosis or damage to the inner. Even when the cell wall is broken, what will happen? All the cellular contents will come out, and that will result in cell death. So that is the mechanism of action of penicillin. I will repeat, penicillin will inhibit transpeptidase enzyme or penicillin binding proteins in gram-positive microorganism. So transpeptidase or penicillin binding proteins is very, very essential for the formation of cross-linking between two peptidoglycan layers, which are made up of N-acetyl uramic acid, N-acetyl glucosamine. Sorry, it is not glutamate, it is glucosamine, N-acetyl glucosamine and N-acetyl uramic acid. Okay, so NaM is N-acetyl uramic acid, where NaG is glucosamine, N-acetyl glucosamine, N-acetyl glucosamine, N-acetyl N-acetyl glucosamine, Glucosamine. So N-acetyl glucosamine. So here what is happening? So there is the peptidoglycan is made up of N-acetyl uramic acid and N-acetyl glucosamine, and both the many peptidoglycan layers are interconnected with each other. So with the help of cross-linking between those two peptidoglycan layers, and that cross-linking is done especially by an enzyme that is called as transpeptidase or penicillin binding proteins. So here penicillin will bind with transpeptidase or penicillin binding proteins. It will inhibit them, thereby cross-linking between N-acetyl glucosamine or N-acetyl uramic acid will not occur, and that will result in synthesis of a bacteria with weak peptidoglycan layer, and finally the cell wall will be weak, and that will result in death of the particular cell. Cell wall will break down, and the inner contents will lose out, and that will result in the death of the bacteria. Okay, so I hope you understand the mechanism. So let me just read it out. So you can see here, peptidoglycan is a major structural component of the cell wall, and that is cross-linked into a net-like structure that surrounds the bacterial cell to provide strength and rigidity. Okay, so this net-like structure, okay, between N-acetyl uramic acid and N-acetyl uramic acid. Here also N-acetyl uramic acid and N-acetyl uramic acid. So between N-acetyl uramic acid, you have a cross-linking here, this one particularly. So this cross-linking will create a net-like structure. So that is very, very essential for the rigidity or strength of the bacteria. Okay, here you can see cross-linking of peptidoglycans is brought about by an enzyme called as transpeptidase. Transpeptidase is responsible for the cross-linking of the cell wall, okay, or cross-linking of the peptidoglycan layer. So here it is clear, penicillin inhibits the enzyme transpeptidase, and so that cross-linking does not take place. So here penicillin inhibits transpeptidase enzyme, here you can see, okay, and this transpeptidase enzyme, inhibition of this transpeptidase enzyme will not allow this cross-linking. This cross-linking will not be formed, am I right? And when this cross-linking is not formed, definitely the bacteria will not survive. Or you can write in this manner even in examination, penicillin binding proteins are bacterial enzymes located on the cell membrane that is responsible for the cross-linking of peptidoglycans. So same, penicillin binding proteins are they are nothing but they are transpeptidase. You can call them transpeptidase or you can call them penicillin binding proteins. So here penicillin binds to the penicillin binding proteins and inactivate them. That transpeptidase or penicillin binding proteins will be inactivated by penicillin, and that will prevent the synthesis and cross-linking of peptidoglycans. Am I right? When there is no cross-linking between peptidoglycan layers, the cell wall deficient bacteria are produced. Means bacteria with a weak cell wall are produced, and that will result in hyperosmosis, swell, burst, and bacteriolysis. Hyperosmosis means, so the water which is present outside the bacteria, it will enter into the cell, okay, and the bacterial cell will swell and it will burst, okay. Is it clear? So a weak cell wall will allow tremendous amount of water or fluids to enter into the bacterial cell from outside, so that the cell wall will have abnormal entry of fluids or water, and the bacteria will swell and it will finally burst, and that will result in bacterial lysis, breakdown of the bacteria or death of the bacteria. That's why initially I use the word penicillins are bactericidal. It will kill the bacteria. Okay, is it clear? Got it? So I will repeat. So penicillin will be very effective on gram-positive bacteria because gram-positive bacteria is made up of peptidoglycan layer. As I told you, these peptidoglycan layers are made up of N-acetyl glucosamine and N-acetyl muramic acid. And here, between this N-acetyl muramic acid on all the multiple layers of gram-positive cell wall, there is a cross-linking which gives strength to the peptidoglycan layer. Okay, and that cross-linking, as I told you, it is made up of L-alanine, D-glutamic acid, diaminopimilic acid, D-alanine, and peptide bond. And this entire amino acids which are cross-linked between N-acetyl muramic acid of one layer and N-acetyl muramic acid of another layer, this cross-linking is done, or this cross-linking is due to an enzyme that is called as transpeptidase or penicillin binding protein. So whatever penicillin will do, penicillin will bind with transpeptidase or penicillin binding proteins. It will inhibit the enzyme. Okay, thereby there is no cross-linking. When there is no cross-linking, peptidoglycan layer, upper layer and lower layer will not be held tightly. Okay, or they become loose, or they lose their structural integrity. They lose their strength. Okay, and the bacteria which is synthesized, it will be deficient of a strong cell wall. Am I right? So the cell wall will not be synthesized properly. Okay, that's why. So the cell will allow large amount of unnecessary fluids into it, and the bacterial cell will swell, and finally it will burst, and that will result in death of the bacterial cell, or what we call it as uh, bacterial lysis. Okay. So now there is a question that has to be explained, because in many books it is not available. Now, if anybody asks you, okay, penicillin mechanism is very clear, but why penicillin is only effective on gram-positive? Okay, why it is very effective on gram-positive? Why penicillin fails to inhibit gram-negative? That is also another question, because in some cases it might be asked in your interviews or in hospitals. Okay, this kind of questions will come across. Why penicillin is effective on gram-positive? Why it is not effective on gram-negative? So this is purely on the basis of its structure. Okay, so here you should understand with what the gram-negative cell is made up of. If they see, penicillin is highly effective on on peptidoglycan layer, and it is going to inhibit the cross-linking between peptidoglycan layers. Okay, so that's why here this gram-positive cell wall, if you observe, it is mainly made up of peptidoglycan. If the same structural arrangement is available even in gram-negative, then penicillin would be very effective on even on gram-negative. But let me show the difference between gram-positive and gram-negative cell wall here. You can see here, please try to understand, this is gram-positive cell wall. You can see here, this is the peptidoglycan layer. Entire structure, entire cell wall structure is made up of peptidoglycan layer, and in between you can see these lines. These lines are nothing but they are cross-linking. Okay, so this cross-linking will be inhibited, and thereby this cross-linking, the enzyme responsible for this cross-linking is transpeptidase or penicillin binding proteins. Penicillin will block this cross-linking, inhibit the enzyme, and cross-linking will not occur. Thereby, the cell wall is formed with weak peptidoglycan layer, and thereby the cell wall collapses. That is what happens in gram-positive cell wall with penicillin. But in case of gram-negative cell wall, please observe very carefully what is the makeup of the cell wall. You can see here gram-negative cell wall is quite different. It is not completely made up of peptidoglycan. You can see here the first layer in gram-negative cell wall is lipoprotein. You can see here first there is a lipoprotein layer, and then there is a peptidoglycan layer. Okay, first there is a lipoprotein layer in gram-negative, then you have a peptidoglycan layer. Like that, there is an alternating lipoprotein and peptidoglycan layer in gram-negative cell wall. Okay, so that's why if penicillin G, first of all, for penicillin G, it is very difficult to cross the lipoprotein layer. Okay, because it has to inhibit the cross-linking. For inhibition of cross-linking, it has to reach the peptidoglycan. Okay, but in gram-negative, so first barrier or obstruction is the outermost layer is the lipoprotein layer. That's why first it has to cross the lipoprotein layer. That is very, very difficult. And even if it crosses, even if it inhibits one peptidoglycan layer, cross-linking between one peptidoglycan layer, okay, so next layer will be again a lipoprotein layer. Next layer will again be a lipoprotein layer. That's why it is very, very difficult. And even I told you, cross-linking will occur only between two peptidoglycan layers. When two peptidoglycan layers arranged continuously, then you have a cross-linking. But here, can you observe any cross-linking? I will say no. Why? Because there is a alternate arrangement in such a way that lipoprotein followed by peptidoglycan, lipoprotein followed by peptidoglycan. That's why there is no chance of cross-linking here. So when there is no chance of cross-linking, can we expect any? Yeah, sorry for the disturbance. So network issue. I will continue. So I think I was showing this particular slide to you. So what I'm saying, in case of gram-positive bacteria, the cell wall is made up of more than 50 layers thickness. Okay, it is made up of peptidoglycan. It is mainly made up of peptidoglycan, and the peptidoglycan is more than 50 layers thickness. Okay, so most 99% of the layer is made up of peptidoglycan. That's why you have a cross-linking, and that cross-linking will be done by transpeptidase or penicillin binding proteins. That's why the target will be clear in gram-positive cell wall. Am I right? So directly they will block transpeptidase, and directly they will inhibit the formation of cross-linking. So if there is no cross-linking, peptidoglycan layers will be very weak. They lose their structural integrity and they collapse. Okay, that will result in the breakdown of bacterial cell wall. Fluids will enter inside, cell will bulge, and cell will burst. So that is what we have seen, bacterial lysis. Okay, whereas in gram-negative, you don't have a cross-linking because there is no continuous continuous formation of peptidoglycan. Okay, so one layer is lipoprotein, then next peptidoglycan, then lipoprotein, then peptidoglycan. Like that, there is an alternate arrangement of lipoprotein followed by a peptidoglycan. That's why penicillin G fails to inhibit gram-negative cell wall. If anybody asks you, this is the answer. Why penicillin G fails to inhibit gram-negative? Because the gram-negative cell wall is not made up of complete peptidoglycan layer in its full thickness. Okay, so it is an alternate. It is made up of alternate layers of peptidoglycan and lipoprotein. That's why it is very difficult to inhibit the peptidoglycan layer, or it is very difficult. Even you can say that there is no cross-linking. Okay, so when peptidoglycan layers are not arranged in series, how can you expect cross-linking? That's why there is no role for these drugs to be done in gram-negative cell wall. Okay, that's why penicillin is highly effective in gram-positive microorganisms, and it fails to inhibit gram-negative microorganisms because gram-negative microorganisms is not fully made up of peptidoglycan. It is made up of alternate peptidoglycan and lipoprotein layers. That's it. Okay. So I will stop now. We will continue in the next coming discussion. Okay. You go through the video. In case you find any difficulty, any doubts, stop me there and let's have a discussion. Okay. Okay. Thank you.